Okay, so for today, we're going to cover the assignment that you guys have about the article reviews. And so at the end of this, I'll demonstrate what kinds of things you should be looking for. But the beginning will all be about how to essentially look at research articles, how to read them, and a little bit about the difference between psychology-based articles that you might find, for example, in EBSCOhost or PsycInfo, and some of the sleep articles that you may find more so in PubMed and sort of medical based. So for the assignment, you guys are going to need to pick your own article about sleep, and you can't choose any of the ones that I've provided you. And this is an example of one that I've provided you and is really heavy neuroscience based. So that way, you guys can see a really good example of something that is an APA format, is not an APA format, isn't an APA format, that is more medical based, and that may be a little bit denser than what you're typically used to in your training. So this particular article was published in the journal Neuron. It is not in APA format, so we're going to kind of walk through where to find things. The first author is Brett Foster, so I'm going to sort of refer to this as the Foster article. And another really important thing is that to always use formal language when talking about articles and papers and studies, and realize that the article doesn't do anything. It's all the authors, right? So the authors conducted the study, the authors said this, et cetera, et cetera. Another really interesting thing about Neuron is that it includes something called highlights. So highlights are really nice because that gives you sort of a bit of information before even walking into the article if this is something that you want to read, because these are the main points and the main findings of this particular article that was published. So this was published in 2015. It was really interesting for many different reasons, which we're going to go into. So this is the intrinsic and task-dependent coupling of neuronal population activity in human parietal cortex. We're going to talk a little bit about what they found. I'm going to talk more of how to read through it than necessarily the article itself. So just to kind of give you a heads up on that. So what are some of the highlights? So they found that human electrocardiography reveals default network regions in parietal cortex. So they found, hey, that there's these particular regions in the parietal cortex that are what we refer to as default networks. They're sort of in place there for humans. And what's really interesting is the word human, and I'll tell you why this is so important in just a minute. They found that the parietal subregions display simultaneous response onset timing, which is really important because it's a coordinated response in terms of timing. They found that parietal connectivity patterns were similar for both rest and task specific and sleep states. This is a really, really, really important point, which is actually one of the main reasons why I wanted to show you guys this particular article for have you to read it for the class. This article is found in the sleeping brain section and is evidence for a particular dreaming theory, and actually is evidence against other very popular theories on dreaming, and I'll explain why in a minute. And then finally, that they found this parietal connectivity patterns were similar for both these electrophysiological measures as well as fMRI. This is important because what this does is it links two methodologies together, finding the same result. So it's using electrical wave measurement on the surface of the brain, matching that up with the fMRI responses that we see when you have somebody sit in an MRI machine with a functional scan going. And so doing functional analyses, seeing what areas of the brain light up or activate, that actually corresponded with the same results that they found for the electrical wave methodology that they used. So let's go into the structure of this particular article. So before we even walk in, before we even look at what's in this article, we already know the main points, which is really nice for the highlights. So here is the main page. So here, summary is actually for Neuron, what we would call the abstract, right? So this gives us kind of the beginning, the middle, the end of the whole paper. This is the main focus of the paper, the sort of a mini introduction, a little bit of the methodology, and also their findings. So we know that it's something to do with functional connectivity. It's something to do with sleep. They're looking at brain networks. They were able to do intracranial electrodes, meaning that in the brain, uh, on the surface of the brain. So they actually took people and they took their skulls off, and they were able to put these electrodes directly on the surface of 3 human subjects. This is really exciting because, as you can imagine, that is extremely invasive. That is a surgery. So we're actually taking human subjects and we are looking at direct electrical activity on the surface of the brain. You see this in animal studies because, ethically speaking, one could argue that, for the sake of science and learning and knowledge, taking a mouse or a rat or even a monkey or a non-human primate, and being able to look at surface electrical activity is better, quote unquote, better ethically than taking a human who is healthy and doesn't have any, health issues, and opening their brain up in surgery and or opening their skull up and looking at their brain. So we don't see this as often. This tells us that something is interesting about these particular 3 human subjects, which we will get to here in a few minutes. So, which may interfere with the findings, and we'll talk about that here in a minute. What they found will be at the end, right? So our findings quantify the spatiotemporal dynamics of parietal cortex during episodic memory retrieval and provide clear neurophysiological correlates of intrinsic and task-dependent functional connectivity in the human brain. So I'm reading this last sentence here. So what they ultimately found was that there are patterns of this particular activity when doing a memory activity. There's these patterns that they found in the brain that correlate with other studies that say, yes, this is where you find this particular activity when you're doing this particular task. When the people were resting, so in a resting state fMRI, right? Uh, uh, and when they were resting for with the electrodes on their brain. So they were able to actually correlate the activity with the same person with fMRI and with the methodology that they use to measure the electrodes. So one thing that I do want to point out is there are a lot of acronyms in this particular article. So here we see AG. I'll just pick that one out. So let's say you're reading through and you keep forgetting. What is AG? One thing that's really interesting is looking at this AG. Okay, well, what does that stand for? Here it's angular gyrus. So maybe you're moving through your article and you keep forgetting what AG stands for. What I highly suggest for you to do is to take that full PDF and throw it into Notability. This is what I have up right now is Notability, and I'm writing all over it, making my own notes, because what I found is a lot of times I'll read an article and there's a really great point that I want to make or want to remember, but if I don't highlight it, if I don't remember it, and you can use the highlighter function up here. So here's highlighter. I've got it set to the pencil, and you can double tap it to change the color. Let's say you want it to be green, right? And you can highlight, maybe you want to highlight, hey, this is really important and I really want to come back to this. Okay. Let's say, oh, you know what? I don't like that. And then you can use the erase button here to erase it. Let's say you want to change the color, you can change it to red because it's super important and you really want yourself to go back and to see this. And then here I'm just going right back to the pencil function. This is really good for you to make your own notes. So let's say you don't remember what this acronym stands for. We'll write it at the top. AG stands for angular gyrus. Boom, now you have it and you can keep going back up to it. Another way to help you with acronyms, if you have a paper that's got tons of acronyms, is to go back to the beginning of the paper. A lot of times in the abstract, they'll use the acronyms for the first time. The very first time they have to use it, they have to write it out in pretty much every format, APA included. If you can't find it there, then you would go to the introduction. If you can't find it there, of course, you can always find it by actually searching the PDF itself. Um, and if you're still can't find it and you're having trouble, you can go to the figure. So the figures on most in most journal formats, they do require you to, if you're going to put the acronym in a figure, that you have to write it out there. So that's also a little shortcut you can run to the figures. Um, so as we're going through this, we've looked at the abstract. That should be the first place that you look. The second place that you look for information is the introduction. And so what I would say when it comes to the introduction, I like to read this not necessarily all word for word. I do a sort of a mixture of like a skim and a good read, just trying to get the information, the background of what they did, especially if I'm not familiar with the particular topic. Some things that they really hit on that's really important that you need to know about this particular study is their use of ECOG, or electrocorticography. Ooh, oh my goodness, my brain and my mouth are just not matching up. ECOG. So they used ECOG to in their methodology to get the electrical recordings on the surface of the brain. And ECOG is a really interesting methodology, and maybe you've never heard of it and you need to know more about it. Well, the introduction section is where you're going to get that information. So it talks about how this information is recorded, and here we learn about the fact that these particular patients that they used, or participants or subjects that they used, and what's typically used for ECOG is those individuals who are going into epilepsy surgery. So if a person has epilepsy and they need to have brain surgery to sever certain connections to stop their seizures from moving one side of the brain to the other, or moving from one part of the brain to the other, if they need a section removed, they will essentially undergo a lot of tests. So they might have the piece of their skull removed and have their brain exposed, and then have certain electrodes placed on for different reasons, so that a person like a surgeon can map out their brain to make sure that they don't do damage when they're doing the surgery. And so what some researchers will take advantage of this and say, hey, let's try to recruit participants in this study, because we have a chance to do this very invasive surgery and invasive methodology on a person who's already going to have their brain exposed. So in this case, and remember, they only have 3 participants. Now, I say only, right? 3 is a big deal when you're considering brain surgery. But in a bigger study, even an fMRI study, you might expect 20, 30, 50, hundreds of participants to be able to get really good information. And here we have 3, which again, for this study is huge. It's not, it's not 1. But it is important for the implications. It's important for the findings. And we'll talk more about that here in a minute. So the information that you get about talking about, for example, resting state fMRI, what is that? What do? You take a person, you put them in the MRI scanner, and then you just have them, sit quietly, or you tell them what task you really wanted them to do, which the task is to be resting and to not physically be doing anything. You want to see, what does the brain do at rest when it doesn't have anything to occupy it, and what does that brain activity look like? So the introduction gives you all this information on all these things that they want to talk about. And so they're going to really emphasize a background of the methodology that they use, the fact that they use 2 different big methodologies, which is ECOG and fMRI. They want you to understand what the research is currently behind those things, and why they felt like what they were going to do was going to find the results that they found. So it's all to support their hypotheses and what they did. And so really, the end of the introduction usually is where they kind of tell you how things tied together, and what they found, and what they were looking for, and what they thought was going to happen, their hypotheses, all that. So the introduction is a really good section to really read through heavily. at some point in your reading. So I like to read through it pretty good, but then I like to go back to it, and I'll explain that in a minute. So if you look here in an APA formatted paper, the next thing you would expect to see is methodology, but here we see results. So because most of you, if not all of you, are trained in psychology to read articles this way, I would say that I would suggest that in an article that says results next, to actually skip it and move down to where the methodology and the method section is, because that's going to give you a little bit better understanding of what these scientists did. So here you say, okay, well, these are all results and they're all, sort of split into sections. Where do we find it? Well, if you keep scrolling, in your paper here, and this is going to go a little blurry. Sometimes it does this if you go too fast and it's not loading. Okay, what you're going to find is if you just take the time to move down, you'll actually find the results section somewhere in the paper. So here, I've scrolled down to page 11 in this particular article, and here we have the experimental procedures. So this tells us exactly what they did. So I would skip the results, and then I would go back to the experimental procedures and say, okay, what did they do? This I would read pretty well. And then after I read what they did, sometimes I like to go back to the introduction just to kind of root down what exactly were they looking for? What did they do? What does the past research say? And move that information all together. So here it tells us, okay, who did they actually test? Well, they had 3 subjects, ages 39, 36, and 22. They had 2 women and a man. And so they had 3 subjects, and that they were going through, they were there for evaluation for neurosurgical treatment of refractory epilepsy. So these individuals were having seizures specifically in certain locations. It actually tells you where the participants were having the seizures, that they all spoke English as a first language, that they were all right-handed, because again, if you want to make sure that your results are going to be consistent across participants, you may want to have them as the same right-handed or left-handed, because we see a little bit of dominance there depending. There's a little bit of conflicting evidence there, but most studies like to look generally at right-handed people. It tells you about how the electrodes were implanted in the specific brain. So each talking about each electrode being exposed and to what size and to where specifically in the brain. It tells you about the specific electrophysiological recording, what they actually did, what that ECOG recording actually was set to, the task that they did. So what was the task that they actually performed when it came to what the ECOG was sort of measuring in terms of brain waves or electrical waves. So here, this explains exactly what the task was. And so that way we can understand, okay, what was the person doing in order for them to for them to make some sort of conclusions about the results of that particular memory task. Here, about the resting state, what did they have the person do? They wanted them to, sit quietly. So here you can say periods of eyes closed, rest. They didn't give them a task. They wanted them to just kind of sit here and do this. And then it also explains sleep data and how that was collected, and how long they collected these periods of data for. The resting state fMRI, how they collected that, they put them in the MRI, and then these are all the specific settings that the MRI was set to, so that way, if you wanted to recreate the study, you could use their sequence and put in all of their measurements and be able to get the same type of scans that they did. Very standard and important for any kind of MRI or fMRI study. Here we can see where they talked about data processing, and they talked about amplitudes and how they did this particular, looking at this particular section of to begin their analysis. They talked about latency and response and fluctuation. They talked about how they clean the data and how they process the data, how they analyze the ECOG and fMRI and compared those together. So this is kind of like what they did, how they prepared the data, And that leads to then what did they find? So here's the methodology. So I would read this really carefully, probably go back to the introduction, make sure I really understand exactly what they did, and then go back through to, in this case, I would skim the results, because if I were you, the more you read, the better you get at it. If you haven't had research methods, this might be a little bit more challenging for you to read. Um, so I would suggest that you probably, skim through something this heavy, skim through the results section, just kind of see if you can get some main points here and there. But I wouldn't spend as much time on the results if it feels very Greek to you, because again, it's something that you need more and more practice with. And if you haven't had the research stats, then it might be a little bit harder for you to process. So what I would do in this article is just kind of skim this section, and it's sectioned out to the most important parts, okay? And then I would then skip to the discussion. So the discussion section is going to really give you what they found, and in this case, in this particular article, which many formats do like authors to do, is to split the discussion up into major sections. So the first part of the discussion is going to give you kind of generally what they found. A lot of times you can look at sort of the last sentence or two, and you can see exactly what they found. So here they're saying they found these similar electrophysiological patterns of connectivity when they did the task, when the participants rested, and when they slept. And so that was really important for the findings of this particular study, because what it does is it demonstrates, hey, when you're sleeping, the things that you were doing throughout the day, they're going to have a certain network that activates in your brain, right? Certain neurons are going to fire when you remember, what did I do yesterday? When you remember, let's play this game, or I watched that TV show, or I did this, right? So these particular memories that you have are going to activate certain networks in your brain. And then when you go to sleep, what this study is showing is, and here are the sort of the 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 actual electrodes placed on the physical brain that show you where the pattern of activity is overlapped with, in the red, you see the red is the fMRI, and the dots are the electrodes that were placed. So it's showing you the overlap between the two methodologies. When you activate these particular networks during the day, the same patterns are seen at night when you sleep. So the brain isn't just resting in the sense that it's doing nothing. What's happening is that the same networks that we see during the day are being repeated at night. And it further strengthens this idea that sleep is for consolidation of memories. It's for the brain to process the memories throughout the day and say, hey, do I want to get rid of this? Do I want to keep that? And so it's reiterating a lot of the evidence, that especially current evidence, that's showing that, hey, our brains are working at night, working while we rest, to concrete certain memories and get rid of other ones. And it's concreting those memories, consolidating those memories by going back over them and going through them, that we see it doing during the day. And so this is a really fascinating study, and a really, really well put together study, to show that what we are finding is corroborates a lot of the previous evidence. So here, going through the discussion, we're picking out the main points, and the discussion is where I'm really going to spend a lot of my time to really understand, what did they find and how did they synthesize that together. So here they sectioned it off here between different parts of what they found, and again, you want to pick out, okay, what were the main points. And at the very, very end, usually, and some articles actually do ask for like a concluding statement or a conclusion. So you will find an actual conclusion, either marked as a section or as a paragraph or a few sentences. Look for this in conclusion. Okay, overarching, what did you find? What were you looking for? So it's going to tell you what the goal is. The goal was to quantify these interactions between this particular area of the brain, looking at task versus sleep versus rest, and compare that with the electrical activity that they're finding through the ECOG, and compare that with fMRI. And if you can get an overlap on the patterns, that tells you that both of those methodologies are doing what they're supposed to, and they're finding the connectivity in the right places, and they're finding the same things, because that's what we want. Is we want to be confident in our results. And if we're finding that both sets of methodologies are saying the same thing, then that must mean that is probably true. Okay. And so here, it also tells us specifically, what can other studies do? Okay. So most papers should include something like implications, like where can we go from here? How can we apply this to the real world? What does it matter? And then here, in this particular case, it should also talk about any kind of limitations. Is it generalizable, right? Is it generalizable across, in this case, they're saying future studies need to figure out, is this generalizable across the whole brain, or across different areas of the brain? Is it generalizable across people who don't have epilepsy, who don't experience seizures? These are all really important questions. So this is just kind of an overview of this particular paper and how I went through and actually read it as a particular exercise. So I'll have two article reviews. One is due before spring break, and one is due before the end of the semester. So you'll have two. So here, this is the one in Moodle. So you're going to need to pick an article that isn't the one that I've given you in either this class or in Sleep and Dreaming if you've taken that class with me. You need to write a review that is not something like the first article I picked, right? It needs to be formal. You need to use language that is, specific to APA format. So in this case, for the Foster article, right? This is Foster et al. Uh, uh, did this, examined this, discovered this, found this, right? In an experiment conducted by Foster et al., 2015, blah, 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 right? So you're going to use formal language. You should have at least 400 words in your review. And my advice to you is to start in Microsoft Word or something like that, and to type it all up and then figure out how many words you have. If you answer all of these questions, you're not going to have any problems getting to 400 words. I promise you, because all of these do require you to really think about this. And what I should be able to do is not even have to really read it to search where did you answer this question. It should be very obvious where the information is as you go through this. So the first thing I want you to do is to explain or describe the actual article, the study, the information that you found. It wasn't an experiment. If it was, what did they do? Very briefly, in maybe 2 sentences, 3 sentences tops. What did they do? What did they find, right? So, in this particular paper, we can say, well, these particular authors conducted a study that was looking at patterns of activation in the brain for while people were resting, while they were sleeping, and while they were doing a memory activity. And they did this with 2 different methodologies, fMRI and ECOG. Remember, if you have acronyms, you've got to write those out. I need to know what those are. Um, and they found that there were similar patterns across all these 3 states of consciousness and states of being. And what does that mean? Well, that means that, the brain is doing similar things in each of these particular modalities. So the second thing, explain the merits of the study or whatever they provided. So maybe it's a review study. What information did they provide? Okay. And what I mean here is, what did they get right? What is some really good things that we can get from this knowledge? Well, what did they do right? Well, they were able to do a methodology here that you don't typically get to see in humans. They were able to actually take human brains and plop on electrodes and see on the surface of the brain what is going on as people do tasks and as people rest and as people sleep. And this is a really great study. They had a lot of merit to it because this is something that is not something that you can do all the time and with everybody. And the information that you get from this can really tell us about things like dream theory. So that the theory that there are lots of different categories of dream theories, lots of different types of dream theories. There are some theories that say, well, your brain is just doing random activation. It's just the neurons are just randomly firing and it's closed off. So your dreams are just an interpretation of random firing. Well, this study that the authors conducted is evidence against that, right? So it contradicts this entire set of dream theories that say that that dreams are just a manifestation, an interpretation of your brain of random firing. These are not random firings. The activation that we see during sleep matches what you do during a memory task, matches what you do during rest. So that tells us that's not random, and that your brain is probably processing information from earlier in the day and figuring out what to consolidate. So there's a whole area of dream research that this dream theory that this actually contradicts and provides evidence for other dream theories that say, no, your brain is consolidating memories and it's functioning and doing things that are important during sleep. And your dreams may be an interpretation of the firings of the neurons during this time, but those firings are not random. So this is a really interesting merits and really good information that came from the study. What did they get wrong? What are the cons? What are the faults of the study? This can be hard. And I know that this for some people can be difficult, but you have to kind of pick through. One place that you can find the authors always have to acknowledge any kind of limitations that they have with their own study. So they have to acknowledge, oh, hey, in this particular case, there are only 3 subjects. There are only 3 participants. Now, in the case of ECOG and human brain directly looking at lectures, that's a lot of people. But in the scope of psychology research, we're expecting to see 50 participants, hundreds, maybe even thousands of participants. If you only have 3, then that could be a problem because we don't know, okay, well, if you only take 3 people out of, a million people, is it going to be the same for everybody? So that's one particular disadvantage of the study. I wouldn't necessarily they got it wrong, but they could have changed that, of course, to make things better by getting more subjects. But again, this, acknowledging that, 3 subjects is a lot for this kind of study. One thing that really is important that we should think about, not necessarily that they got wrong, but one thing that you can say that they're very confident in their findings. The people that they used had epilepsy, they had seizures. This is not a neurotypical group. This is not a group of people that you can say represent all of people, all of humans, because they have a condition. So you can say, well, their results aren't generalizable to the public, to the main population, potentially because they have this condition. So that's another thing that you have to think about. Okay, well, what is something that they could have fixed? Well, obviously, in a perfect world, you could take a person who doesn't have anything wrong with them and look at their brain. But of course, that would mean that that's a very invasive and non-ethical, procedure, because they don't need their brain opened, and that would be incredibly invasive. So, but some of the faults, some of the cons, some of the limitations of the work you can talk about here. So it may not necessarily be a problem or something they did wrong, but it might just be a limitation of the study in of itself. Um, or maybe you find something that they did wrong, something that they either do or don't acknowledge, and you read through and you say, oh man, they should have done XYZ instead of that. So maybe there were mistakes made that you can pick out. The final thing that I want you to talk about in your article review is the implications of the information that they give. Okay, what are they implying? How can you take this and apply it to everyday life? Is this a public health concern? Should people be concerned? Should they be given this information? Should this be an article that's provided to everybody to learn about the brain and sleep? And so in this particular really interesting article, the implications could be, well, now we're finding that the brain is doing certain things during the day, the same as the night. So one of the implications is that you can say, okay, well, now let's revisit all these dream theories, right? So that's something that we can find useful. Another thing, if you want to look at it as sort of like what to tell the public, what to tell, Joe Schmo on the street. Well, your brain is so important and sleep is so vital for you to do the things that you need to do every day. And so one huge public health message is if your brain is doing all these memory things at night while you sleep, then you need to make time to sleep, because if you want to have a healthy brain and healthy memories and be able to function, whether you're a college student and you're worried about academic performance, whether you're, got a really important job where if you don't remember something, like maybe you're a surgeon and you don't remember something that could kill someone, you know, or if you're just everyday person, doing everyday things, you want to have a healthy brain, and you want to have a good memory. And in order to do that, you need to give yourself the time to sleep. So some of the implications of this is just simply you need to make time for sleep, because sleep is important, because your brain is doing really important things at night while you sleep. So what you're going to do is you're going to, give all that information. I should be able to see everything that you have gone through and understood. And then you're going to include the word count from Microsoft Word or whatever it is that you use, of how many words are in your review. And it should be over 400, minimum 400 words. But if by answering all these questions, there's no doubt you'll be able to get to 400. 400 is about a small paragraph. Try to do everything in APA format, and then give me the citation at the end of the paper, or I should say the reference at the end of the paper. You can cite throughout your review. And then you're going to then upload the full text, the PDF, one PDF per article review on here, so that I can look through and see, okay, what did you find, and actually read the article myself to make sure that it lines up. So if you have any questions, to email me. And hopefully this kind of helped you work through a little bit more difficult article than you're probably used to, and also give you a little bit of a head start on the sleeping brain section and this particular article that you'll be reading. So if you have any questions, let me